Well, this is surprising. Out of all the advancements I would have expected would be made, being able to restore the capacity to learn and cellular activity in brain tissue after it's been cryogenically preserved was just not one of them. Now, people are running with this in all sorts of interesting directions, including just the capacity to cryopreserve a brain, as well as being able to upload a brain to a computer, and we will talk about it. It gets weird. I do want to preface this and say that these are not necessarily realistic applications, but we're a step closer to them, and I would not have thought this paper was possible. Researchers decided to focus their investigation on the hippocampus. This is the part of your brain that's responsible for learning and memory. If any part of it could demonstrate the capacity to learn, it would be that part. Now, they did a couple of different kinds of experiments. One of them involved taking just brain tissue from the hippocampus of a mouse. The other one was in situ, so inside of the skull of a mouse, and that did present some additional challenges. They replaced the blood with a cryoprotectant and then flash froze the tissue and kept it frozen between 1 and 8 days. They revived it, took the cryoprotectant out, and put it in a medium that would resemble cerebral spinal fluid. And for the in situ one, they took the hippocampus from the rest of the tissue and put that in the spinal fluid. They found that yes, not only was it able to maintain spontaneous electrical activity, but it was capable of learning. Now, the method they used is not dissimilar to our little brain friends. Yes, the brain organoids, the tiny human brains you can grow in a literal jar if you want to. You see, they can be hooked up to electronics. It needs to have a two-way street, where you can communicate with the little brains and then they can respond. The responses they give indicated that they are able to change their electrical pattern based on their input, meaning they retain the capacity to learn, reinforce pathways, and make new pathways. That is incredible. This means that the capacity to learn and grow is still there after they've been frozen. Now, some would like to apply this to reviving brains after they've been cryopreserved, and I think that's less realistic than uploading your mind to a computer, to be perfectly honest. Neural activity is not the same thing as function, and your brain only really functions in the context of having everything else around it. Well, yes, you can grow a brain outside of a skull. That's not the function of an adult brain. It's a miniaturized version. But this is a step closer to that goal, I guess. More than likely, this is going to be applied to neuroscience as a new way to preserve samples rather than any radical medical application. But why do people think that this applies to uploading your mind? Well, that has a little bit to do with this paper. Researchers were able to model an entire mouse brain. Yes, all 26 billion synapses and 9 million artificial neurons. That means the spiking activity of every single individual neuron was displayed. In fact, they could even communicate with each other. But some of the comments that one of the authors has been saying leaves pause. I think it is entirely possible for a piece of hardware to be a thinking, feeling entity. I really don't know how to say his name correctly, but you can look him up. The argument is that if we can construct an entire mouse's brain in a virtual world, if we can run every single spiking neuron and watch it learn and grow, perhaps we could get it to be conscious. But if we could do that, then you could scan a person's brain and create a digital copy of it. Now, the kind of scale that is required to run a mouse's brain is basically an entire warehouse worth of supercomputer. Researchers are trying to do the very same thing with a human brain. If they can do that and create an AI of sorts that's based on neuromorphic computation with every single neuron in a brain working, what is the difference between that and our brains? If we accomplish this as humanity, we might just find out. The reason that people want to apply this to cryopreservation of brains is that if we can preserve the neural activity, perhaps we can scan it and upload it. Rather than trying to get a cryopreserved brain back online, perhaps we can turn it into an AI. This is not real immortality, because if you copy yourself and 2 or 3 or 10 of you can all exist at the same time, you have not achieved immortality. You have simply copied yourself at best, and at worst, created a suffering intelligence. I don't know what's going to happen next. It will be interesting, and I hope it leads to robots having rights. Follow for more.